Dr. Fernandez is an assistant professor at the Allen School for Global Health, and as a disease ecologist, her research focuses on the eco-epidemiology of zoonotic diseases, and in particular, vector-borne diseases. Her main research interests lay in understanding the transmission of zoonotic diseases as complex socio-ecological systems, combining methods from epidemiology and ecology. The ultimate goal is to identify critical factors affecting disease transmission, which will aid in the design of improved intervention strategies to alleviate the disease burden in affected communities. So I'm really happy to welcome today Dr. Pilar Fernandez. So Pilar, I'll hand it over to you.
Thank you. Thank you for the introduction and the invitation, and thank you for the video. It's great to see the final product. So I'm going to go ahead and share my screen. Go. Okay, so everybody can see it, right?
Yeah, it looks good.
I'm going to have the laser, and I'm going to hide this window so I don't see myself. Okay, so actually the video was a great introduction to start talking about zoonotic diseases and how we're studying them and why we're talking about integrating the social aspects with the ecological aspects and what is the framework that I use to study diseases. Before I move forward, I always like to start the talks with a little bit of an introduction of where I'm coming from, what's my trajectory, because I think it really matters to understand the projects that I've been working on and how I'm approaching them. So I'm originally from Argentina. I did my PhD, all my studies, undergrad, grad, and PhD at the University of Buenos Aires, where I was working on Chagas disease. I finished my dissertation in 2017, and then I got a fellowship to do my postdoctoral work at the Earth Institute at Columbia University. And the Earth Institute is an interdisciplinary research, so that's when, even though at the PhD level, my work was very interdisciplinary, this is where I think it consolidated as an interdisciplinary scholar. And then in 2020, September 2020, great timing, I started my appointment as an assistant professor at the Allen School for Global Health, which it's a great fit because it's also very interdisciplinary and has allowed me to expand my research and integrate different disciplines into the type of work that I do. So just to give an overview of the framework and what is eco-epidemiology. When we think about zoonotic diseases, and we have gone through the COVID-19 pandemic, we have seen an outbreak of Mpox that has spread globally. And those are great examples, very recent examples of how spillover events from wildlife reservoirs can impact humans in a very great way. And I think those outbreaks, we can all agree that are happening more and more often for many different reasons. And one is how humans are interacting with the wild environment and how we're modifying it with our activities. And one of the things that I wanted to bring to your attention was, and this is a great review that actually happened, it came out a couple of years ago, but this is an analogy of how our spillover events are what we talk about when one pathogen that it's circulating in wildlife or domestic animals spill over into humans, or there's cross transmission between species. And that's what we call a spillover event. A spillover event can lead to an outbreak, which can expand and lead to a pandemic, for example, in the case of COVID-19, or it can be contain. And we really think that the spillover events are happening very, very frequently, more than we can actually measure. But what leads to an outbreak and what the outbreak actually leads into a larger epidemic or pandemic, that are different scales of analysis. But looking at this kind of Swiss cheese analogy, there's a lot of things that need to happen from the wildlife reservoir for a disease to a pathogen to jump into humans and be able to infect humans. And we can study all this different layers of complexity from how it's circulating in wildlife, and how are humans interacting with the environment that leads to that interaction and exposure, to actually being able to trespass that cross-species barrier and actually lead to infection. So there's a lot of disciplines, and you can see it in this slide, that are actually studying. And usually what we have been doing traditionally is we have to study all the systems separately. So animal epidemiology, veterinarians have been dealing with animal epidemiology, so they have studied how transmission occurs in animals of commercial importance. In agriculture, humans have, like, human epidemiology has traditionally been separated from all these other components. And in animal ecology, or more traditionally disease ecology, we've just been focusing more on what's happening in wildlife. So the idea is to think about integrating all these components. In classical epidemiology, as we think about in animal epidemiology, but also in human epidemiology, we're just focused on like these three questions: is what is the distribution of disease in a population, what causes a disease, and what's the impact of disease. But when we bring ecology, basically ecology is the interaction, and we study the interactions and the relationships between the different organisms. So we can think about host and pathogens interacting. If we have different hosts, we can consider humans and host, how are humans interacting with other animals, or other animals are in, like different animal species are interacting between each other, and how the environment is shaping those interactions. So we can have coexistence, but we're also thinking about the environment and what is allowing that infection to happen. So that's what ecology really brings to the question. So it's not about how it's distributed, but going a step forward and really understanding how those interactions happen. So in sum, ecological epidemiology, which is the framework that I use in my studies, is the study of the ecology of infectious diseases. So it goes from the population level studies, which are traditionally in epidemiology, to bring community level studies, to think about those interactions. And by community, I mean the different species that are circulating, including humans, and how those interactions really shape how the pathogens are transmitted. And also we can think about including the environment. Now, when we talk about zoonotic diseases, there's a lot of focus on the ecological and biological factors that affect transmission. So how are the species interacting together, and what are the competence of those hosts to acquire infection and transmit it back. But we usually neglect the socioeconomic and cultural factors that kind of shaping that transmission context. And we tend to, again, study those separately because we don't think they're domains of human health. And now I think it's more and more obvious of how the context really affect that transmission. So when we think about global change, we can think about that environment that I mentioned that kind of shapes these interactions. We can focus on the natural environment, but we can also think about the built environment and how the humans' activities are changing both. Because when we think about global change, we're not just talking about climate change, but also how our activities, like for example, agricultural activities, really shape that environment and changes those interactions between the hosts, where they are, how many they are, and how they interact to allow transmission. So putting all this together, in my lab, what we're doing is we're studying zoonotic diseases, trying to integrate all the pieces of information. And to integrate all the pieces of information, we have started using this framework that was originally developed in physics, which is basically trying to think that just studying the parts independently, it's not enough, because in systems, we have emerging components. So we really need to understand how and map out how the whole system works to really understand those complex interactions that might emerge when we study the whole system together. So we're trying to integrate the human and ecological components to understand not only the emergence of new diseases, but also the persistence of zoonotic diseases, because we have newly emerging diseases, but we have diseases that have been around for a long time. So we're trying to focus, and that's basically why my research is interdisciplinary, because even though I'm an ecologist by training, I've been working a lot with anthropologists, sociologists, and economists to really bring this social context into the research and really integrate it in as another component. So what we're trying to do is not just study how many events were there happening and try to anticipate patterns, but really understanding this underlying system and trying to map out how they are. And so with that, we can really understand what are the leverage points where we can actually go and act to reduce the burden of disease, but also what are the unintended consequences that might emerge by doing one or another intervention. So in the types of methods that we use, from the video I mentioned, we do a lot of the fieldwork component. It's really key in the research. We do animal samplings, we do zero surveys in humans for different projects. We also use traditionally epidemiological tools like knowledge, attitudes, and practice surveys to really understand the human context and the practices that people have, and why, really understanding also going a step beyond and really understanding why are people behaving the way they behave. And so we can adjust for those changes. Obviously, because we're working with communities, we believe that community engagement is key to work on this system. So we have we try to develop a really strong relationship with the communities where we're working, just to make sure that we communicate and they are on board with the research. Because in my experience, if we don't have that, then the whole research fails. And also you can get some insights from the people because they are the ones that are affected and they're more closely related. So there's a lot of the knowledge that we can leverage in our research. And then we come with all this data and we use mathematical modeling and statistical modeling, so quantitative analysis, to really understand. And some of the examples that we have done is on doing spatial analysis and risk mapping to really map out where the risk is. And so we can have a pragmatic tool to deliver interventions. We have used some mathematical models to try to gain some insights on how the system works. And then obviously we have done a statistical inference from the data to really understand what is the association between all these risk factors. So with that, I think it's obvious that my work is very interdisciplinary. And as I mentioned, I work with collaborators in other fields and other disciplines. And I see myself as translating between disciplines. So sometimes I think, and I wanted to bring this to your attention, this is a piece that we brought when I was a postdoc at Columbia University, part of that cohort of the Earth at the Earth Institute. Is like, what are the, I think we all agree that interdisciplinary research and cross communications is important, but sometimes it's hard to achieve because there's so many barriers in our disciplines that allow us from really going beyond and really achieving that integration. I've been really lucky that I'm at the Allen School because I think that's really value, but I think there's, we still face some challenges with funding agencies. NSF has done terrific work trying to really acknowledge this, and there's like new calls where the interdisciplinary approach is really, really value. But we're still face other challenges. So I just wanted to bring this to your attention. If you want to know more about and discuss it more, I'm happy to talk about. This is something that I really feel very passionate and how universities can support interdisciplinary work. So our projects are based in, we have projects in South America, particularly right now I have 2 active projects in Argentina, and I have an active project in the Palouse, which is the video that you saw, that where we're focusing on rodent-borne diseases and tick-borne diseases as well. So hantavirus, rickettsia, Rocky Mountain spotted fever, those are examples of pathogens that we're trying to monitor here. So with that, I'm gonna focus on 2 quick stories. I just wanna make sure that I have some time for questions, but I think I wanted to kind of give an example of how this framework has been used. So my first story is about Chagas disease, which is a vector-borne disease that it's transmitted by this bug that it's called triatomine. It goes from northern Argentina to southern US, but it's a really, really big problem in Latin America as a parasitic disease. In the areas where I'm working, which is this region, the Gran Chaco region, it's one of the 10 global hotspots for neglected tropical diseases, where we're really trying to understand how all these land use changes are affecting the transmission of diseases. And we have newly emerging diseases that are affecting these communities that usually impoverishment. And I also wanted to kind of bring to your attention why we do global work. And sometimes I think the importance, what we're struggling with a lot, I think in global health in general, is that we fail to recognize that focusing on the transmission, and Chagas disease has been around for many, many years, still a problem in endemic areas. And because people move and emigrate, it has become a problem in other regions. So it has become a problem in Europe, where they don't have the vector, but it also has become a problem in the US through migration. And you have potential vectors here, especially in the southern part of the country. And most of the people, still 60% of infected people worldwide, live in that Gran Chaco region that I mentioned. So from there, transmission is still really high, and transmission persists worldwide through human movement. So if we really can control infection in endemic areas, we'll have this persistent problem of zoonotic diseases being exported and becoming a problem. So where do we need to focus? Do we need to wait until it gets to the countries, or we really need to focus on where it's really happening and try to reduce the burden of disease there? So another example, so I started working with this disease for my PhD, and I still been engaged, and now we're taking it to a new stage, but I just wanted to bring some of the attention to some of the results that we have had for that region. So I'm working here in northern Argentina. And the interesting thing about this Chagas disease in this region is that this is usually a native territory, which has a very, very recent history of colonization. So they started to show their hunter-gatherers people, and the sedentary patterns really started at the early the 20th century, when with the first settlements for Creole, which is the people that are descendants from European people, and started becoming them. And so they used to move all the time. And this vector, the characteristic of this vector is that it really needs to stay there for a long time to really achieve high abundance. And so because of the constant movement, they didn't give that opportunity. And so this is a picture from the early 900s. And you can see that it was very precarious, but they were moving all the time. With the first Creole settlements, they started changing into this sedentary patterns where the houses are still really precarious, but they were not moving as much. So that's where Chagas disease started becoming a really big problem, because people were not moving their houses. It allowed to achieve really high abundance of the vector, and transmission start occurring really in the domestic environment. And this vectors is really plastic, so adapted really, really well to human dwellings. And you can see in the 90s, still, you can see this houses were mud houses with thatched roofs. And this is recent pictures that I took. Well, now they're like 10 years older, but you can see this, you still see mud houses and some government plannings, but Chagas disease is still a problem. So when I was talking about mapping out the system, this is what I was talking about. So we made a comprehensive map of all the evidence that we have and hypotheses that we wanted to test. And particularly, we wanted to really understand how the social determinants of health affected domestic transmission and human infection. So the first challenge we had is like, how do we measure socioeconomic status in rural settings that are very impoverished? We cannot use any income. And because if we measure that, all households were the same. But when in our observations from the field, we could see that not everybody not all the households had the same resources. So we took this, I worked with economists to develop the social vulnerability index, which took into account a lot of other, the multi-dimension diseases of poverty. So from demographic to education to actually what they have in terms of livestock. There's a lot of trade. And then informal work and some monetary income in terms of welfare. So we use that, and on the other side, we wanted to see is, okay, how is it affecting the interactions, those interactions that I mentioned, and we can measure those interactions by looking, for example, in blood feeding vectors. What's the proportion of blood meals that are fed on the different animals? And in this case, those vectors feed on any vertebrate that are around, and in those domestic environments, dogs, chickens, and humans are the main hosts that they feed on. They mostly feed on humans, but you can see there's a lot of cross transmission, and these different colors here mean that they're from the same bag. We detected multiple feedings, and dogs, particularly in the systems, are really, really important because they are the main domestic reservoir. So if a vector feeds on a dog, that means they have really high parasitemias and they're going to get infected, and so they keep the cycle going on, and it's a really high risk for humans. But we observe that if we increase the number of as the number of bugs increase in a household, the number of triatomines that fed on humans decrease, and the number of animals that the number of bugs that fed on dogs increase. So there's like that in kind of trade-off between, it's like, yes, there's more infectious bugs around because there's more contact with the dogs, but they're diverting in a way a little bit away from the humans. But still, overall, they are infecting, it's a risk factor for humans. And we wanted to see, and this is where we brought the social vulnerability. This is overall, and just wanted to see if was the same picture in the houses with high social vulnerability and low social vulnerability. So high social vulnerability means that they don't have a lot of resources to deal with, and they are very precarious houses, and they have less resources to deal with infection and infestation in their households. And we found that in low social vulnerability index, remember these are really poor houses still, but we saw this, that this trend between the vector abundance increasing, contact with the dogs, really depending on how many bugs you have in the house. But when we look at high social vulnerability houses, so the extreme, the poor of the poor, what we observe is that this relationship between vector abundance and dogs was not really the same. So it was independent. So what it means is that we don't need that many vectors to have really high contact rates. And this can be for many reasons, and where are the dogs sleeping in the house? Where is the micro distribution of vectors in the house? And so there's a lot of questions that we want to explore of what explains that even if you don't have that many vectors, you still have high risk because they're still contacting a lot of dogs. And again, dogs are the main reservoirs that maintain the transmission in the domestic cycle. So it's still a high risk factor for humans. So then we wanted to look at human infection. So for that, we work in these communities. We do sero surveys on 80% of the population there, around 2,000 people, a little bit more. And we run our model integrating a lot of this information that we've got at household level. And again, we observe, and I'm not going to go into the details, but I want to bring to your attention this negative interaction that we found between the social vulnerability and the infected vector abundance, because that's where we, the same idea, how we interpret this is that we don't need that many vectors in a house to really understand that still they have the high risk. So people living in poor houses with not that many vectors are still at high risk of infection because of that contact between the dogs and the vectors that maintain the transmission. So with that, this is an example of how we can translate into an actionable tool to direct transmission. And why is this important? Vector control for Chagas disease usually is focused on spraying, insecticide spraying in the human dwellings. And it's usually the decision-making of that is based on where the vectors are and how many there are. So if you want to prioritize areas, you focus on the areas where there are vectors. But basically, we show by integrating the social vulnerability, we can actually find those households that otherwise would be completely neglected by if we just focus on vector density, and they will not probably be sprayed or not considered as high risk. But we've by integrating these two components, we have found that they're really high risk. So I also wanted to mention really quickly, I'm not going to go, I can talk a lot more, but I also focus on tick-borne diseases. And there's a lot of, this is more local, this is worldwide. Ticks had transmitted a lot of diseases, and this is an example of some of the ticks. I don't need to tell you that tick-borne diseases are increasing. Most of the people know that. And so we have the same idea behind it. We wanted to study if more ticks bring more cases. So we're starting incorporating the human dimension, and we kind of map out. I'm going to go really quickly over this, but we're using agent-based models, so mathematical models, to really understand the human tick contact rates and how they happen, and how the environment and the fragmentation levels that we observe in the landscape really affects the interactions between humans and ticks, but really focusing on also incorporating the human behavioral components and how people make decisions on how the interventions happen. So that's something that's happening right now. So sorry, I kind of rushed through a lot of things, but I was very optimistic with time. But yeah, happy to take questions.
Thanks, Pilar. That was really good. I have a question, but I'll let others go first. Anybody else have a question? Okay, I loved it. And I watched the video way too many times as they were editing it, but that, you know, when you say where the magic happens, and it's clear from your presentation today and in that video that the magic for you is when you're working with community humans and, you know, you're going and talking to them. So I'm just curious if you can talk to us a little bit about how you establish those relationships of trust, you know, to get the data that you want that's going to be helpful, you know, and, you know, how, and what is that magic for you? Does it mean you actually go in a different direction with your research, or it opens up something new, or, you know, what is that?
Yeah, no, that's a great question. And I think it's an undervalued part of research that we usually tend to neglect because we're not trained to work with communities. So it's something that I've learned from trial and error. But I think it's when we studied, especially in human disease, we really need to understand who is affected. People have a lot of knowledge about, like, empirical knowledge. They have been affected, so they can tell you where it's happening. Like, from their perspective, right? And from us, our job is to bring the science and really try to kind of dissect those observations that people have and really understand that, but go back and forth. So I think part of, and I think an important part of the research that we do, especially if we're gonna work and get information from people, is to really understand what are the motivations, what is their priorities, and really explain what are the outcomes, like what's our perspective on the problem from a scientific point of view, and we can some shed light on some of the questions that they have. So I think it's an important part that needs to happen before we start doing research, but along the way, and really have a two-way communication and really be open to what people are saying.
Oh, that's awesome. Chris, it looks like you have a question.
Yeah, quick question, Pilar. Thank you. Great talk. You mentioned agent-based models at the end. Are those things typically in your field? Do people write them themselves, or are there a few big models that everybody uses, or how does that work?
So yeah, again, this is not it's been used more and more, but it's recently we don't have a lot of tradition in our field of using agent-based models. They have been using it in economy for decision making, in other disciplines. So there are some really big models with synthetic populations that have been used and they're being developed. But I try to I use this mathematical models. Usually I develop them just to answer specific questions that we have, and that they're addressing the system. Because one of the problems that we have with mathematical models, and especially with agent-based models, because they're so complex, is how to validate them. So I feel like there has to be very specific for each of the problems. And what I tell my students is like, really understand what your question is and you design that model to answer your question. But yeah, they're being more and more used these days for infectious diseases.